I'm happy to introduce today's seminar speakers. Lydia Heasley, for today's seminar. Lydia received her PhD in Michael McMurray's lab at the University of Colorado Medical Campus at Anschutz. I'm just going to call it the medical campus. She did her postdoctoral, very independent postdoctoral work on genome stability with Luis Argueso at Colorado State University, and got hired as an assistant professor back in her alma mater, her PhD alma mater, the Department of Biochemistry and Molecular Genetics at the University of Colorado Medical School campus. She's been there only since 2022, so we are hearing a seminar from a young, fresh, obvious Colorado ski person. I can tell that right away. Throughout her scientific career, Lydia's been interested in the structure and stability of eukaryotic chromosomes, an interest that's near and dear to my heart as well. She's recipient of an NIH Pathway to Independence Award that built upon her discovery that bursts of genomic instability can cause complex multi-chromosomal aneuploidy and other structural variations, challenging sort of conventional ideas that this kind of genomic evolution requires more progressive subsequent cell divisions that can happen very quickly. And so her data provide clear direct evidence that cells can quickly achieve highly altered genomes. And this has really interesting implications for phenotypic stability and plasticity. She also has interest in the same kind of genomic elements I'm interested in, such as the RNA repeats, which can change genetically very fast within a short number of generations and have phenotypic impacts. I invited Lydia out for a genetics seminar because my lab really enjoyed her thoughtful seminar that she presented at a FASTA meeting we attended in 2022. And I really thought her fundamental interests were perfect for coming to the genetics Laboratory of Genetics and giving a seminar here, because there's several colleagues within this sphere that the Laboratory of Genetics that would really enjoy hearing her talk and getting to meet her. And I hope she's had a really good day. She's been a pleasure to host. She's very, very nice young woman. Graduated from high school in 2008, which really freaks me out. Like I can't even. I don't even want to go there. But I'm looking forward to hearing her full seminar about her research and the direction she's taking her new lab. So with that, let Lydia get started.
Thank you very much. And thank you for hosting me. I'm very happy to come out here and see everybody in Madison. And I'm going to stay around here, and seems to be a glitch between my computer and the projector. So if my mouse is in the wrong place, I'll try and look up here, but please feel free to interrupt if I'm talking about something that you're not seeing, because it could just be something going on. Okay, so let's talk about structural variations in the genome. Okay, so Darwin famously described evolution as a slow, iterative, stepwise machine, driven by small, minute changes and a very kind of short or long-term and long process. And after years of studying evolution and variation over time, we now know that these short and small successive variations are driven primarily by genomic mutations. And most of the work to this date has been done on point mutations or single nucleotide variations in the genome. And what we've learned from this work is that indeed mutation rates may differ between species and organisms, but mutations are acquired in a linear fashion at constant rates over very long periods of time. But there is another class of variation and mutation in the genome, collectively known as structural variations. And these types of mutations, by their very nature, defy a lot of the principles that Darwin laid out in The Origin of Species about how evolution occurs. And structural variations come in many different flavors, but what unites them all is their regionally acting mutations. They can come in the form of full chromosome gains or losses, called aneuploidies, which I'll focus on a lot today. They can occur as segmental copy number variations that lead to the duplication or deletions of segments of chromosomes in the genome. There's the very big class of chromosomal rearrangements, which occur through many mechanisms and have differential impacts on the integrity and subsequent structure of the genome. And then there's more debatably inconsequential types of structural variations that are often copy number neutral, but impact the heterozygosity of the genome through mitotic recombination between homologs. These are disparate, and they each belong to their own, family of mutation, but structural variations have been somewhat arbitrarily designated as mutations that impact more than 1 kilobase of sequence. But in fact, these structural variations at all classes can impact megabases of DNA in a single moment. And so in that sense, they impact many orders of magnitude more genetic material than all point mutations probably combined in the lifetime of an organism. Even before we knew that DNA was the hereditary material, pioneering geneticists like Barbara McClintock and Dub Zansky and Thomas Hunt Morgan recognized the importance of structural variations as being important sources not only of genetic diversity, but also phenotypic plasticity. And the types of mutations that these guys were looking at under the microscope were the enormous classes of structural variations, because they could actually see them by light microscopy, like this chromosomal inversion on a chromosome of the fruit fly Drosophila pseudoobscura. And while these are a minority of the large structural variations that you can discern by eye, these actually help to solidify gene theory, because for the first time, geneticists and molecular biologists could actually see a change in chromosome structure and associate it with a phenotype, an organismal phenotype. And herein lies the paradox that has plagued structural variation biology for a long time. We've known about these types of mutations for longer than any other, over 100 years, and yet we still understand very little about what causes them and what impacts they have on organisms. So why is this? Well, the short answer is that basically everything that makes structural variations so interesting makes them very hard to study using traditional genetic techniques and approaches. Structural variations are hard to define at the genetic level because they often arise through recombination processes at repetitive elements. And so they resist traditional mapping techniques like PCR across breakpoints. And they've been difficult to find through other modes of kind of mutation identification. And they're often multigenic. They involve multiple genes because they're so big. And so linkage analysis is difficult with structural variations that affect many genes all at once. Their consequences are also multivalent because of their size and the way that they reorganize the genome. And I'm showing all of these potential consequences as kind of amorphous blobs here because there's so much pleiotropy between these consequences. So a single structural variation can impact the gene dosage and regulation at a local level. It can impact how that chromosome orients in space within the nucleus and have extend all the way up to phenotype or to consequences on the three-dimensional organization of the genome within the nucleus. And each one of these changes can have an effect that's difficult to separate from other causative effects stemming from the same mutation. And so these two challenges have historically made it very difficult to understand how variations arise in the genome and how they go on to influence biological processes and phenotypes. And because of these challenges, the incredible amount of phenotypic diversity observed within eukaryotic populations has been attributed to the types of mutations that we can understand, like point mutations. And so for a very long time, structural variations were thought to be both incredibly rare, but also very detrimental to the cell because we didn't see them very often. In the last couple decades, however, this has changed. The advent of new genomic approaches like short and long read sequencing and the host of bioinformatic techniques that we can now use to probe and mine these sequencing data sets have enabled us to not only see structural variations with new clarity, but to identify many new structural variation classes that we didn't know existed. Collectively, our new lens with which to look at the genome allows geneticists and genomicists alike to take a more holistic perspective on what genome biology means. And what we can all kind of agree on now is that the genome is much more than the sum of just its parts. And this is an important perspective that we need moving forward to understand how structural variations are acquired, and then starting to parse apart those blobs of consequences to really get at the mechanisms that drive each consequence of a structural variation in the genome. I just show this little graph to tell you that if you PubMed search structural variation, this is kind of the plot of the number of publications relative to the same search for point mutation. And so I just put this in here to highlight that we're learning a lot right now, but collectively this field is still in its infancy, which I find very exciting as a new professor, but also it just means that there's questions we haven't even gotten to ask yet because we haven't even thought to ask them. And so there's a lot of really interesting biology to come our way. But what have we learned in the last couple of years? The most exciting thing is that the genetic data and the ability to sequence genomes and look at them with clarity now has demonstrated that although we don't know what they're doing and how they all got there, structural variations actually import more genomic diversity between individuals in a population on a per kb basis than all single nucleotide polymorphisms combined. To the limited extent that we understand all this ubiquitous structural variation, it's clear that structural variations will have, and currently have, and we'll probably discover more, links to human health that are very important. So structural variation in an uncontrolled manner is a hallmark of tumorigenesis. And we're also discovering how certain structural variations lead to the development of both neurological and congenital genetic disorders. But we don't yet have a handle on how structural variation is controlled or the dynamics with which it occurs in germline, developmental, and somatic contexts. And this makes it very difficult yet to understand the ways in which these types of genetic mutations impact a patient and the way in which the penetrance of these disorders manifests in the clinic. But we've also learned that structural variations are probably important and dominant capacitors for phenotypic plasticity and adaptation. They're not always bad. In fact, they're probably very important to natural populations for the ability to change. And I'll get into that a little bit in my talk. But again, we have a lot to learn here too. And important first questions that my group is really interested in understanding is understanding, first of all, what the composition of structural variations is between individuals within a population and how this reservoir of potential variation impacts the variation at the phenotypic and population level. So it seems right now that the more we look for structural variations, the more we find. And again, I find this really interesting and exciting because it shows us just how much more we have to learn about the eukaryotic genome. More importantly, though, it argues that until we can incorporate structural variation and the mechanisms driving structural variations in the genome into our theories of evolution and mutagenesis, we're not actually going to have a good idea of the forces that keep the genome intact or allow it to change over time. And so my new lab is building on postdoctoral work I did before starting. And we're thinking about three big questions in the field that I'm going to touch on a little bit today. The first is, with what patterns and dynamics does the structure of the genome evolve? How does genome evolution modulate phenotypic potential? And how do structural variations shape the architectures of populations and species over time? And I'm going to breeze through these just because I don't want to go over time, but hopefully I can make it through and tell you a little bit about all three of the progress on these. For all of the work that I'm going to tell you today and all of the work that's going on in our lab, we use Saccharomyces. I think I'll go through this especially fast because I don't think I have to convince anybody in the audience how important of a model organism this is for genetics. But basically, we use it because it's the best understood genome and cell, to my knowledge. But what makes it such a powerful system for our purposes is that it's a complex yet small and tidy little genome. It possesses all of the features that larger, higher metazoan genomes do, but it's small, which makes it easy to work with. And because almost everything we know about the genome stability pathways that are conserved from yeast to humans were actually discovered in yeast. Cerevisiae is still an exquisite model for genetic selection, and with it we can design elegant tools to recover spontaneous mutants out of populations. Because the genome is so small, we can sequence many clones at very deep coverage for very little cost. So we can sequence individual yeast genomes for about $15 or $20 now, which makes us enables us to have some robustness in our sequencing data. It's not just descriptive, it can get at mechanism too. And then one of my favorite things that you'll see quite a lot through the talk is that the yeast genome is organized such that we can actually resolve intact yeast chromosomes through agarose gels. And this is a pulse field gel. These are not PCR bands. These are telomere to telomere length chromosomes. And you can see the structural variations with your own eye on a gel, which is nice when we're taking survey level basically looks at the diversity across populations, but it's also an important confirmation step for any of the structural variations we deduce using sequencing approaches. Also, I don't have to touch on this too much, but it's becoming well appreciated that Saccharomyces is a really powerful system for understanding evolutionary genomics. I think a lot of people think that budding yeast is just a poor hobbled microbe that lives a sad life on a laboratory bench, but in fact, it's got a very rich natural life, right? And our reliance on it for industrial processes and brewing and baking for millennia we have a very intimate relationship with this species, and we've done a good job of curating a lot of natural isolates. And we know a lot about kind of the SNP-based differences between these isolates, but nobody's started to look at the diversity of genome structures across the species. And I see this as a really important untapped resource. Just to give you a sense of this, here's a pulse field gel like I just described of just a handful of wild isolates of Saccharomyces. These differ extensively at the sequence level, up to 1%, but I hope you can appreciate, if you take nothing away from this talk, besides this, the amount of structural diversity that exists between individuals that all fall into the same species. And these may be millions of years separated or something. They're quite divergent, we know that. But we still don't know what most of this structural variation is, what the basis is, which is really cool. But if we want to take an even shorter time scale, here are 7 clones from the exact same population, 7 colonies I picked off of the same streak in the lab, and they have structural variations that differ from one another. And the difference is that at the sequence level, within the body of these chromosomes, they're identical. And yet structural variations that occur in the repetitive regions of the genome occur so often that you can almost detect them on a per cycle, their changes on a per cell cycle basis. So this is just how dynamic these parts of the genome are that we are just now starting to look at. Okay, so I'm gonna start in on some of the other stuff, but if I lose you along the way, the things to take home today are that structural variations do not follow neo-Darwinian paradigms of acquisition. They tend to occur in bursts, and that they have really dramatic impacts on the expression of environmental stress responses and phenotypic plasticity in wild populations. Okay, so again, kind of the Darwinian paradigms. I started the talk with kind of mandate that evolution or mutations are acquired linearly, randomly, sequentially over time. And we know this is true of point mutations, but many cell types across eukarya harbor really remarkably complex structural variations and structurally variant karyotypes. And they not only evolve these, but they maintain them for long periods of time. And it's remained unclear what the basis of this, what the dynamics of that evolution might look like. The best example, of course, are probably that we're familiar with are the karyotypes of human solid tumors. By the time they're detected, human solid tumors are remarkably altered by structural variations. And I'm highlighting many that we can see at the gross level, like by eye. Many of these are aneuploidies, which we'll talk about today. Aneuploidies, again, are whole chromosome gains and losses, and they're thought to arise primarily by segregation errors during mitosis when the chromosomes segregate each cell cycle. In the context that you're probably most familiar, they've always been marketed as really deleterious mutations, but I think it's important to remember that these are readily exploited adaptive mutations in microbial populations, and if you guys like beer, or wine, actually, most of the yeasts that were involved in that process are highly aneuploid and they're exquisitely fit in their natural environments. So the way that the question was to try and understand, and I'm not going to present all of the data, but to try and understand how, by what, is aneuploidy acquisition a stepwise process, or does it occur with a different dynamic? Do we see multiple aneuploidies arise simultaneously, or is it truly a Darwinian stepwise process? And to do that, we use heterozygous diploid strains that we've engineered with selectable cassettes. So we take 2 haploid strains that differ greatly at the sequence level. They're still in the same species, but they have tons of single nucleotide polymorphisms across the chromosomes. This helps us immensely when we sequence them. So we can see loss of heterozygosity, copy number variations, and assign it to each individual molecules, right? But for one of the parents, before we make these diploids, we integrate counter-selectable markers onto each side of a centromere of a given chromosome. And so what we can do is make this diploid. We can grow cells for very limited cell divisions, very small populations, and plate those populations onto selectable media. And cells that retain this marked chromosome will die on that selected plate, but cells that lost that chromosome will form a colony on the plate, and we can recover the aneuploid clones in the population. We can do quantitative fluctuation tests to calculate rates of missegregation or rates of loss, but we can also take those clones, prepare DNA, and send it for sequencing and see what the genome looks like across the board. And that's the data I'll present right now. This is the way that I'll kind of graphically depict the yeast genome for the rest of the talk. We always work with diploids, and Cerevisiae has 16 chromosomes. So the yellow bars each denote one homolog, and the black bar denotes the other homolog of a given chromosome, and the amplitude changes will denote changes in copy number. And so when we select aneuploid clones for a given chromosome, we've done this for a handful of chromosomes at this point, and we pick them randomly from plates and we sequence them, we get 3 defined flavors of aneuploid karyotypes that crop up. Very often we see karyotypes in which only the chromosome that bore the cassettes is lost. So just a monosomic strain that's lost one chromosome spontaneously. But nearly as often, we recover clones whose karyotypes have been tremendously altered by structural variations, and almost always aneuploidies. So if you select for an aneuploidy, the accompanying unselected mutations that come along for the ride are also aneuploidies. These come in a couple of interesting flavors that I'll touch on in a minute, but I hope you can appreciate just how much structural variation can arise in a single limited generation experiment. And we've actually gotten to the point where we're quite confident that these probably all arose in a single mitotic cell division. To determine if it was even possible that these karyotypes down here could actually form through a Darwinian independent process, we modeled the number of generations it would take, given the known rates of chromosome missegregation in the yeast, for the yeast organism, the number of generations it would take to acquire the types of karyotypes shown in this figure. And except for the case where only a single chromosome is missegregated, every single other chromosomal karyotype that we observed would require more generations to form than our experiment allowed. We would never have recovered these in the population if it had been a sequential acquisition type of dynamic. And we did some other experiments to show that this isn't a mutator phenotype in which you first lose one chromosome and then you lose other chromosomes very quickly, that these are probably instantaneous bursts-like events. We named this phenomenon systemic genomic instability, which I like less and less every day because it's such a mouthful. So I'm going to refer to them as bursts for the rest of the talk. But effectively, this kind of harkens to a very famous model of punctuated equilibrium that was put forth by Niles Elbridge and Stephen Jay Gould that postulated maybe that every once in a while in a phase of stasis, a crisis occurs. And for some reason, cells become uniquely vulnerable to genetic instability, and in this case, chromosome loss or chromosome gain. And in that moment, they acquire many new mutations all at once. But after that, they go back to a basal rate. And I'll talk about this more in a minute, but I repeat this over and over again when I give this talk, that these are wild type cells grown in very rich conditions. They're not stressed, they're not selected, besides for mutations that arose in the population stochastically. So this is kind of just an inherent part of genomic instability that we're trying to figure out. So altogether, we found that gradual and burst-like modes can alter the genomic landscape of a cell, but these bursts occur frequently, more than we ever thought, and they can drive the rapid formation of profoundly altered architectures of the genome. They also indicate that there's a shared mechanism, because if you select for aneuploidy, you get more aneuploidy. We've done similar experiments. If you select for loss of heterozygosity, you end up getting extra unselected loss of heterozygosity, and this kind of points us towards the molecular mechanisms that might be underlying these events. And then finally, again, these occur stochastically in unperturbed genetically normal cells. So the new lab is taking some of these ideas and starting to dig into a mechanistic basis. I really want to know what causes each one of these. And so again, what we found are some distinct flavors of aneuploidization that occurs normally in eukaryotic populations. One flavor that affects just an individual chromosome. A very interesting flavor in which the homologs, both homologs of a given chromosome, are missegregated simultaneously, leading to whole chromosome loss of heterozygosity. It's not called uniparental disomy. And I'd love to talk to anybody else about this afterwards, but I don't have time to today. And then the bursts, in which multiple chromosomes are simultaneously missegregated. And this is really interesting to me because I kind of thought bursts would be the main thing because chromosome segregation is such a global process. So this is a video that I took in my first postdoc of a kangaroo rat cell going through mitosis. And you can see that the entire cell has devoted itself to partitioning the genetic material with perfect fidelity. So what happens when these processes go wrong? These processes that survey the entire cell, that keep track of all the chromosomes. How do you end up with different flavors of aneuploidy randomly in a population? And we get some first hints from the work that I did as a postdoc, in which we found that the relative frequencies of these different flavors of aneuploidy differed on a per chromosome basis. So what I'm showing here are kind of the relative frequencies of bursts and monosomes and that cool uniparental disome for a handful of chromosomes that we performed that selection assay with. And what you can see is the frequencies of each one of these, each flavor of these events actually differs on a per chromosome basis. And what that suggests to us, and we have some preliminary data to back up, is that there are features unique to each of these chromosomes that predispose them for different types of instability. And so work that we're doing in the lab now is starting to go through systematically and figure out what those elements are and the mechanism by which they destabilize chromosomes. And we have, we're thinking about things like whether different centromeres function better or worse than others, the composition of transposable elements and content on a chromosome, special genetic regions like the rDNA array, and even the size and architecture of telomeres on different chromosomes. We also have some fun evidence that some of the stochasticity of these events arises from just cell-wide changes, fluctuations that don't originate at a specific chromosomal locus, but are actually just a freak moment in a cell's life. We're thinking about noisy gene expression being a dominant cause of this, perhaps cellular age, and we found some interesting new connections between kind of the activity and health of the mitochondria and the resulting stability of the chromosomes in the nucleus. A second thing that we're thinking about that I've hinted at a couple times is that the existence of such bursts of variation adds a new and important layer of complexity to kind of our current paradigms of genomic instability. And moving forward, our goal is to start to think about what does genetic stability really mean on a per cell basis and over the lifetime of an organism? Is it as static of a thing as we've always thought? I think I always learned genomic instability was a chronic disease state, not that it could be a transient fleeting state. And so starting to understand the basis of these types of patterns of genomic change and understanding whether genomic stability in real time is more of a periodic or a fluid, flexible state. And we're doing some fun experiments to get at that. I'm going to switch gears, and it's going to seem like a really harsh transition, but I promise it's going to come back full circle by the end of the talk. I wanted to, especially with this audience, talk a little bit about how we've been using natural yeast isolates to understand, first of all, what structural diversity looks like across a population, but also to understand what the consequences of such structural variation might be for populations. And so, yeah, early in my postdoc, I wanted to start to understand what structural variation looks like between people or between individuals in a population. I work with yeast, not people. Genomes are too big. But again, I bring this pulse field gel up just to remind you of the crazy amount of structural diversity that does exist between individuals within the same species, and to kind of shout out that we can see this on a gel and it's spectacular, but we still don't know what the basis of most of these structural variations are. And it turns out that they are remarkably complex and types of rearrangements that we didn't necessarily think would be conducive with fitness and life. And so this is really fun work that the postdoc who just joined my lab has picked up and is making really great progress on. But anyways, when John McCusker, a pioneering yeast geneticist at Duke, retired, he sent me his collection in the mail, and so I have a wonderful and very honored to be the custodian of that collection. But I have all these wild isolates in the freezer that we can use now to start to ask these questions about natural diversity. And just to highlight kind of the techniques and some of the observations we've made on just a single strain, I'm going to use this one strain, YJM311, which is here in the second lane, as a kind of a proof of principle for some of the work that we're doing in the lab now. So just some background. YJM311 is actually an opportunistic isolate that was recovered from a human patient as an infection, a systemic infection, by John McCusker in the 90s during the AIDS epidemic. Cerevisiae can be opportunistic pathogens, especially in the context of immunocompromised patients. And that trend is growing, unfortunately, as most fungal pathogens are growing too. But anyways, it's got some really interesting phenotypes that are associated with that virulent potential. From a genomic standpoint, it's a diploid. It's highly heterozygous, both at the SNP level, but you can see, well, maybe you can appreciate this because I haven't done a good job explaining pulse fields, but there's tons of structural diversity between the homologs in this. So it's not just sequence diverse, it's structurally diverse as well. And it's been known for a long time, mysteriously, that there's a mutations in this background that basically kill all the spores that it makes after meiosis. So when fungal cells go into meiosis, they can make spores, which are basically the sperm and eggs. And this is an important route that we're still trying to figure out its importance in the evolutionary life history of the species, but being able to make gametes and do sexual reproduction is generally an important part of eukaryotic life cycles. So the fact that these spores are dead on arrival is kind of closes the door on a very important part of the strain's life cycle. But we didn't know what it was, and I kind of wanted to understand it. But unfortunately, at the time, there was no phased data for the structural variations that existed in those genomes. They were just bands on a gel. And we had the short read sequencing for it, but that's very difficult to use for resolving structural variations. And we also didn't have it phased by parents. So when you want to just think about structural variations and their impacts for an organism or an individual, you need to be able to resolve them and assign them to the two homologs of a given chromosome. You don't want those reads mixed together because it will be confounding. And we didn't have any of this for YJM311, nor for any other diploid strain of Saccharomyces. And so during the pandemic, I worked on the computer a lot and came up with a pipeline to take long reads produced from nanopore sequencing and turn them into completely resolved genome assemblies. And we did this for YJM311. And this is what the genome looks like at the cartoon level. There's entirely too much to talk about here. This could have been the whole talk. But I'll just summarize some of the amazing things that we were able to see in this single genome using long read sequencing. We were able to resolve the remarkably diverse and plastic structures of the telomeres and the subtelomeric elements, see how these change from cell cycle to cell cycle between individuals. We were able to phase transposable elements, not only to see where they're at in the genome, but to understand how they facilitate continued evolution of that genome over time. And then we were able to find some really crazy structural variations in the genome of YJM311 that actually have important consequences for its life history and evolutionary trajectory. We were also able to see really cool horizontal gene transfer events that I'm not going to talk about today. But in fact, the structural variation that seems to be the cause of the spore inviolability that I told you about was the fact that one of the homologs of chromosome 1 had actually circularized and become a ring chromosome, while the other one stayed as a linear molecule. And this has no impact on the fitness of that strain in mitotic growth. It grows like crazy and it's very fit. But it's a big deal during meiosis because when you do meiotic crossing over between a circular and a linear molecule, you almost always get a dicentric chromosome, which when it's segregated during anaphase 1 and 2 ends up as a broken chromosome. And so most spores that are produced from this strain actually end up with an incomplete complement of the genetic material and can't germinate. And so this has provoked some interesting and exciting ideas that we're thinking about how structural variations could be drivers of intraspecies isolation and population divergence. And just as a hint of what we're thinking about, it turns out that most of the strains on this gel have very, very, very poor spore viability. and complex spore inviabilities that aren't just explained by a single point mutation that's recessive or something like that. So we think that within the Saccharomyces species, there's actually tons of diversification and isolation going on, and we're hoping to use some of these techniques to get a handle on that. When we were working with YJM311, we also made this really exciting observation that that this strain, unlike laboratory strains that don't do anything fun anymore, can make these really crazy phenotypic changes. And so, as an example, this strain, YJM311, normally forms nice smooth colonies on YPD plates like you're used to seeing in yeast. But about 1 in 1,000 cells would develop this really crazy, ornate, lacy phenotype. And we didn't really know what this was, but it was a spontaneous variation in the population. And the more we thought about it, the more it looked like kind of the variant colony architectures that wild populations of yeast cells can turn on in a programmed way. So normally many wild yeast strains, not all of them, but when they sense a change, an acute change in their environment, like the depletion of carbon, like a sugar depletion or something, will turn on a stress response pathway in which they go from a unicellular growth phase to kind of a cooperative, communal, almost multicellular growth phase. And they can form these really lacy, ornate colonies. Within these colonies, cells are specified for different fates. Some are shunted towards doing meiosis and they make spores and they hunker down. Others switch their growth form and they start to scavenge for nutrients. Others start to adhere and stick to each other to form kind of a biofilm or to invade the matrix below them. They really, we don't have to have the debate, but by most metrics, they check the box of being multicellular in this phase of growth. And this is believed to help them to weather environmental stresses until better times come along. But our strain is growing on YPD, and still some colonies, some cells within that population have decided to turn this program on. Why? Why would they be doing that? And the literature said that it was probably a new point mutation, but we knew that the genes or the pathway that controls this response is very complex. It involves 10 to 15 genes that are dispersed across chromosomes in these genomes. So SNPs or a single new point mutation didn't quite fit, but we decided to take a bunch of these and a bunch of these and sequence them and see what we got. And whereas the wild type clones, like the normal smooth ones, had absolutely no variations or point mutations in the genome, every single ruffled colony that we sequenced had new variations, and they were all structural variations, not a single new point mutation within the bunch. And by and large, these were aneuploidies, and they were most often chromosome gains. We did detect a few clones that had manifested this phenotype through a loss of heterozygosity event, and a couple rearrangements, chromosome rearrangements, but these chromosome rearrangements were also resulting in copy number variations. And so what this taught us is that this stochastic phenotypic switch is driven by copy number variations, and most often these copy number variations are acquired through whole chromosome gains. And here's kind of the graph, those little graphs showing you the karyotypes. And what I hope you can appreciate is that very few of these karyotypes have just one chromosomal gain. Most of them actually had multiple chromosomes gained and lost. And so this helped us to verify that, or this was kind of encouraging because it showed that bursts not only occur in wild populations of yeast, but they are dominant modes of variation when we're talking about the expression of very complex traits. I was encouraged to see this, and I was excited not only because I think these colonies are super cool and I would really like to continue following up on the biology of them, but this highlights the strength of working with the wild yeast strains beyond their interesting genomes. Like I mentioned, most of the lab strains we work with, we've bred these cool phenotypes out, because if you had a strain that was switching from smooth to rough in different conditions, it would make them very hard to work with. You can put these through a flow cytometer, it would make them hard to get single cells. So we've kind of bred these qualities out of our lab strains. But what we can actually appreciate now is that structural variations that arise don't just have a silent impact on the cells that experience them. In fact, they can have really dramatic phenotypic consequences to those cells. And I also want to touch on on one thing, I pulled this straight from my job talk, but I'm going to go through it anyways. That these two approaches that we take in the lab of engineering a strain specifically in the lab for selection and for sequencing analysis, and using wild strains to look at phenotypic changes are really complementary. Because when we engineer a strain like we did in the first half of my talk, we're getting exactly what we want, and we're able to apply a very quantitative analysis to mutation and to de novo structural variation. But by necessity, in building a good selection system, you're saying, give me only what I want. And so we miss most of the structural variations that probably arise in a clonal population of cells. But when we work with wild strains, or when we look at phenotypic changes, just spontaneously arising changes in the morphology of colonies, for example, this takes a lot of work because you're screening tens and tens and tens and tens of plates looking for a few spontaneous variants that pop up on the plate, right? So this is more of a screening approach, not a selection approach. But in this sense, we're saying, give me everything that does happen that matters, or that generates a consequence, right? And so through both approaches, we're able to find that this burst-like pattern of structural variation not only exists, but it drives relevant phenotypic changes. And we don't know how relevant those are in nature yet. That's a hard question to tackle, especially in the laboratory, but we're looking at some other phenotypes like antifungal drug resistance, and I can tell you that the same principles seem to apply for the acquisition of antifungal drug resistance in Cerevisiae at least. So altogether, what we're learning and starting to appreciate in the new lab is that structural variations, and specifically, it seems right now, aneuploidies can potentiate phenotypic heterogeneity in very small clonal populations. These types of mutations arise quite rapidly, and we're starting to ask questions of why cells would be okay with that much aneuploidy, but it seems like if it allows them to adapt quickly, it might be okay. But we also see multiple patterns of structural variation. So there's different clocks ticking within the same genome about how and where in the genome structural variations arise. And collectively, this kind of burst-like phenomenon, or just the acquisition of new structural variations, introduces phenotypic diversity into a population, karyotypic diversity, but then the stability of those genomes after they've acquired new structural variations also modulates the frequency of switching and of continued evolution. And I didn't talk about this too much, but many of these ruffled colonies will switch back to the smooth state, and this is dependent on the stability of the genome. And so the dynamics of that, we're still starting to parse out and understand if there's some rules that we should know about that switching frequency to predict kind of the stability of the genome overall. So I think I maybe breezed through. Oh, not too bad. Hopefully I didn't go too quick, sorry. I hope that I've convinced you that structural variations are kind of this new and exciting part of the genome that's always been there, but we're just now starting to see. And by accepting that these are parts of the genome, we're having to recontextualize a lot of our notions of what genome variation and stability actually look like. And with that, I just want to quickly thank everybody in the new lab, which is small. I'd also like to thank the folks in my postdoctoral lab, in Lucas's lab, especially Hyatt Vincent, who was a very talented undergraduate who did a lot of the selection plating that hasn't been published yet, but it will be published soon. And she was a great friend when I was in the lab. And my new home, or my new old home, back in biochemistry and molecular genetics, the NIH for the funding, and I'm happy to take any questions.
Curious whether you saw anything like obvious attenuation of like growth rate with some strains that just had stochastic aneuploidy. Because you mentioned that, I mean, a bunch of these colonies in criminal populations develop aneuploidy. So is there anything obvious on the plate that
Yeah, totally. Yeah, not for all of the chromosomes, but there's usually the aneuploid, like the cell that's lost a chromosome makes a smaller colony in the same amount of time as a euploid cell, but that's actually not fully penetrant across chromosomes. So for example, cells that have lost a copy of chromosome 3 grow almost as well, pretty much just as well as cells that have 2 copies of chromosome 3. Whereas cells that have lost a copy of chromosome 9 grow much slower than cells that are diploid or disomic for chromosome 9. It doesn't necessarily scale with the size of that chromosome though. So what I think is it could be a little bit more complicated than just like more genes, more consequences. But yeah, there's definitely phenotypes. Yeah. Yes.
What is the time interval that you measure between the periodic bursts and basically millions of genes?
Right. So when we do the normal selection assays, we're growing cells from the time that they're a single cell on the dish until they're a 2-day-old colony on the plate. So that's about 30 generations. But we've also used sectoring assays to show that you can account for all of the chromosome missegregation events, basically, in a single event. So in a sectoring assay, you use a color marker or some kind of phenotypic marker to get both halves of that cell division, and then you can see all of the aneuploidies. And this has been done in the past too, and similar things were observed. Yeah, so it's not always one cell division, but it quite often is. And what we've found is sometimes if you happen to inherit a very unstable karyotype in that burst, you will be unstable. And so we've taken some of these spontaneous aneuploid clones, and we've done mutation accumulation experiments on them for outwards of like 250 or 300 generations. Some of them change immediately, and sometimes they adopt an even crazier karyotype, and then they're stable for like 300 generations. And then there are others that are born with a very crazy, like what I consider a crazy karyotype, and they're stable from then on out under neutral selections, just about bottleneck passaging. Thank you.
Really cool stuff. So you specifically were showing these examples for one of the strains that you had, and you were seeing this kind of prevalent phenotype even on YP growth. I'm curious if you have seen this and found this in other strains, and then also if you can test this by putting like actually purposely trying to stress them and see if you see the darkness showing up at a higher rate or see the same kind of circle variation.
Yeah, those are great questions. So the first one is yes, lots of the wild strains we've looked at will do the switch. The way you can induce the switch is just to take cells that were growing on YPD and grow them on YP low dextrose plates that have 0.5% dextrose instead of 2% dextrose, and they will all make the Lacey phenotype. But if you plate clonal populations, small colonies like I showed you, of these strains onto YP low dextrose plates, at the same frequency, you get smooth colonies. So you get 1 in 1,000 cells will be unable to do the induced switch now. And those are driven by the same flavors and structural variations, different chromosomes, but there's what we think is that there's activators and repressors of this pathway. And if you change the dosage of the copy number of those upstream factors relative to the rest of the pathway, you can either lock cells into one state or the other, but you get the variance occurring in both the induced and uninduced states. It's just yeah, good question.
And just to follow up on that, because this clarification, so the low glucose is the signal. I was wondering if you can elaborate a little bit on that. Like we grow our yeast in the sun, right? It's the control situation is like going to the Grand Hotel with the star.
Yeah, right.
And I was wondering, just because of some anecdotal experiments we've done in our lab, if that may actually promote this kind of effect.
Life is just too rich.
No, it's not too rich. It's just that maybe like here micro, maybe under those situations, it's kind of like life is good. Let's just try some shit, right? It's like
no, that's a great point. Yeah, I love that, and I think that this is there's yeah, I love this idea, and we've been talking about it a lot. That maybe for microbial populations, the basal resting state of a microbial population is heterogeneity, where you're just throwing off hopeful monsters and freaks because you're hedging your bets that it's not always going to be good. Testing bet hedging is fraught, but I like to use that. I see it more as a bet hedging mechanism, and it's been proposed as such in the past. And that way you have all the variation, you have all the options created so that if times get really bad, there's probably a survivor subpopulation within that group that can kind of carry the population through. And the beautiful thing about aneuploidies is that they're revertible. So if by gaining a chromosome, you're able to survive a stress, I don't know, a raindrop hitting the colony, or being put in a context of fluconazole where you need to be resistant to it in order to survive, having an extra copy of a chromosome might get you through that because it'll be selected for at that point, but then you can always go back to the normal genome. So I think that we know that Saccharomyces cerevisiae prefers to be diploid, although I know there's people in the audience who know better about that than I do. But, you know, we think about the fixed karyotype of organisms, and that's probably true in the context that we've characterized them, like on a YPD plate. But the importance of transient karyotype changes in natural settings, even if they're fleeting moments in the life of an organism, I think is a very under thought.
They evolve to evolve.
Right.
Right.
Yeah. And then, so whether the fidelity of mitosis is, the infidelity of mitosis, I should say, is under selection is something I think about, but I don't let myself think about too much. But I really like the idea that,
I like that idea.
yeah, that when everything is plentiful, this is the time to kind of hedge your bets that things won't always last.
In our microbes, so that was selected for. We came from the microbes, and now right. It's all their fault.
Yeah.
That's cool.
Okay.
I'm skeptical that non-destruction could be positively selected, but it's an interesting thing.
Yeah, I am too.
My question is kind of related to this. So you've got a whole bunch of diversity of essentially mutation events rarely happening. It's really exciting to see all those. But then thinking about the kind of standing variation in this kind of organism, if we weren't considering clinical cases or industrial applications, would we really see this much karyotypic variation? And have you tried looking at the kind of karyotype variation compared with the rotational variation? My guess is that a lot of it goes away, right? That it appears by mutation, but then doesn't persist over a long period of time. And I'm trying to think of why that, if you think that's right, why that might be. The udifferential disomy seems obvious, is that a structural variation has occurred, but it's not one that actually changes karyotype per se.
Mhm.
And then the other thing I thought could be a factor is what you mentioned about sex, which is if you're highly aneuploid, doing meiosis is now a lot harder. So do you think there's kind of a way to get at what selection might be doing with all this variation over the long term, like comparing these kinds of data sets?
There probably is, but I don't know how to go about doing it. I think that it's hard to say how fleeting I mean, I'm talking about aneuploidies, which are like the monsters of the structural variation world. There are other structural variations that are permanent. They occur, they arise, and they're stuck in the genome for as long as that piece of DNA is around. You know, they're not nearly as plastic as aneuploidies. But I think that I always suspect that we might lose some information when we take a strain out of its environment and put it on a YPD plate to grow it up and sequence it. And that if aneuploidies really are as fleeting and kind of ethereal as they seem to be, that we may be retrieving strains from the wild that are aneuploid, but we lose that information by the time that we've made DNA out of them and sequenced it. I don't know how to get around that, because until they decide to do single cell genome sequencing for yeast, which doesn't seem like anybody's interested in doing. But yeah, I mean, so the colonies, I did like a back of the envelope calculation, and aneuploidies are probably the most fitness impacting structural variations that we can think of. And about 2.5% of a colony is aneuploid for at least one chromosome. So it's a non-trivial percentage of a population, but yet we don't pull those guys out very often when we do other types of experiments. So yeah, I think a lot of it is probably a dead end, and it's only under selective pressure that you might find the benefit in those. Did that answer your question? No.
Yeah, like it's like some combination of these factors, like reversing the aneuploidy and possibly some direct selection.
Yeah.
And then some indirect selection with the sex stuff. I mean,
Right.
Something like sex is a whole other can of worms, but
Totally.
it's been maintained in this organism and most others, presumably because it has some benefit, and losing that ability by being aneuploid should be over the long run, you would expect it to be detrimental.
Right. You would think, yeah. But I do think that,
Transience seems like,
yeah, I think that's where it's probably most important. But I think that you know, the trend for spore inviability because of structural variations, we've only looked at that in the context of clinical isolates, which are kind of monsters, right? I mean, who knows where they're forming? They're usually very mosaic. They come from they're hybrids, interspecies hybrids of divergent lineages. They're genetically unstable to some extent, and so it's hard to know has that strain, how old is that strain, and will it ever have to do sex to survive? You know, maybe it doesn't, and so maybe having a structural variation like that just has no consequence in its short-term and maybe long-term life trajectory. Yeah.
I have a few questions here from Zoom.
Oh.
Live, they're pretty straightforward, so I think we're gonna get moving really fast. First one is when the disease goes back to the screen, is this correlated with further mutations?
In the cases that we've looked at, the phenotypic reversion from rough to smooth colony is usually accompanied with the reacquisition of the chromosome if it was lost, or the loss of the chromosome if it was gained. So it usually results in not point mutations, but just going back to the diploid karyotype.
Clonal packages are very restricted to biofilm formations. Are all the clinicals that are listed in the same isolates looked at once with one is developed?
No, not necessarily. In fact, the thing that's kind of plagued the opportunistic strains of Saccharomyces field for a while, and just the premise that they could be opportunists, is that there's no uniting phenotype that they all share. They don't all make biofilms. They're not all temperature resistant. They don't all flocculate. So yeah, no, it's not a complete rule.
Was it only one strain you were seeing the test continuously going on? So do you have any ideas what this strain is switch?
Wait, I missed the first half of that question.
Let me just give a question. So what strain are we thinking? It's like number 3.
Oh, yeah. So the question is this YJM311 the only strain that spontaneously switches from smooth to rough? No, we've seen others that also spontaneously switch. And actually so switching, looking at morphology switching is really cool, but again, you have to plate cells at such a dilute concentration that you have to screen hundreds of plates. But we've actually found that spontaneous fluconazole resistance is a quicker way of looking at what is probably a relevant phenotype, but is driven probably by structural variations, and lots of wild strains will throw resistant colonies spontaneously. Yeah.
The question based somewhat off of Daniel asked, this idea of standing variation. If you did this kind of experiment in a completely homozygous diploid, do you expect that you'd find the same frequency of aneuploid formation? Because now you lose allele-specific parts.
Totally. That's a great question. We have done that. Where we marked the same chromosomes with counter-selectable markers and selected for their loss in a homozygous background. And the rate was lower, but the rates of spontaneous missegregation and bursts were lower. But there's some caveats to that experiment. The first is that we did it in the laboratory strain, which I think is probably sensitized to aneuploidy, so it might just not be tolerant enough to carry them and make colonies. But the other one is that we use different counter-selectable markers. So for the homozygous background experiments, we used UR3 as the counter-selectable marker. In the stuff I talked about today, we used cannabinine. And there's a different recovery rate. So there's a lag time with UR3 counter selection that will eliminate about 10% of the clones that actually are true UR minus. So I think that I wouldn't be surprised if the rate is lower, but we haven't carefully done that experiment. But one thing we've been thinking about, the graduate student Ginny in my lab has been found that the intraspecies hybrids of Cerevisiae that are naturally occurring, most of which have been pulled out of the clinic, they are venom sensitive inherently. So we think that maybe these intra-species hybrids that come from, that have divergent backgrounds that might have significant allelic differences, not only in expression of genes, but in like the function or the compatibility of gene products and protein complexes, could actually be inherently unstable. Just because there's enough diversity within them that they're a little bit unstable.
Are you gonna get nanopore sequences for the wild strains and use some kind of phylogenetics across cycles?
Yep. Yeah, we're doing it right now. Matthew joined the lab as a postdoc, and that's what he's decided he wants to do. And so, yeah, we're going to generate nanopore sequencing for a large swath of natural strains or wild isolates, and then the cool thing is like we can look at some of these structural variations are so dynamic. Like the things that are going on at telomeres will be very hard to assign any kind of phylogenetic designation to, but one great example of how some structural variations can actually be used as like dates of relatedness are the horizontal gene transfer events. And so we can see these in these wild genomes that they're in exactly the same orientation, the exact same locus between the same two base pairs. And that means that these strains that we thought were very distantly related actually are very much more likely to be related. And then these sequences, these horizontal gene transfer events, don't really function that much in the Cereviceae genome, so they degenerate over time. And so you can actually date independent horizontal gene transfer events and look at kind of how long they may have been there relative to other strains. So I'm really excited to see what that looks like. Good question. Okay, thank you so much.